This is young Optimus Prime and Megatron.
I may not have a cog, but my finger can transform. Guess which one? I'll give you a limited option.
Excuse me, sir. Allow me. I apologize on his behalf for the disrespect.
However, this is Optimus Prime and Megatron after equipping their energon cores.
Nice.
Before Optimus Prime and Megatron become the legends we know, they were just ordinary bots. They lost the fights a lot. But after getting their energon cores, they became real Transformers, and everything changed. They have more power, more resilience. I believe most boys have dreamed of being the kind of hero who comes back stronger with a cheat engine. That dream definitely stuck with me. As a kid, I started to wonder, what if we could bring that kind of technology into real life? To even begin answering that question, we have to first talk about DNA. DNA is like the blueprint of life. It holds the instructions for how living things grow and function. This molecule has a special double-headed shape and is made of four types of building blocks: adenine, thymine, cytosine, and guanine. These spaces paired up in a specific way. So we have A with T and C with J, creating kind of code. And that code will tell the cells how to make proteins. Proteins are basically the tools that do everything inside a cell. They help build structures from chemical reactions, carry signals, and more. To get from DNA to a working protein, the cell first copies the DNA into messenger RNA. This step is called transcription. Then, that RNA is used to build the protein, and that step is called translation. Altogether, this process, going from DNA to RNA to protein, is known as the central dogma of molecular biology. But even if a gene has a perfect code, it will not work unless the cell knows where to start, what to read, or when to stop. That's where regulatory sequences come in. Promoters tell the cell where to begin copying. Ribosome binding sites help guide the machinery that builds proteins. Stop signals mark the end. Without these, a gene is like a site with no title, no instructions, and no ending. Now having the basic background information about DNA, our own energon core could be considered as DNA segment. Meanwhile, Frederick Griffith's experiment in 1928 provides powerful support for our thoughts. Griffith was working with two types of ammonia bacteria. The first type was called the smooth strain, and it was deadly. The other type, called the rough strain, was harmless. When Griffith killed the deadly bacteria with heat and mixed them with the harmless ones, something strange happened. The harmless bacteria transformed into killers, and when scientists went deeper, they found out the reason was from DNA. It was the first clear evidence that DNA carries genetic information. Based on this experiment, a genetic editing targeted DNA engineering technique was gradually developed. This technique has an interesting name, transformation. Scientists use it to give bacteria new traits on purpose. The secret weapon is something called a plasmid. A plasmid is a small, circular piece of DNA that bacteria naturally carry. It's like a bonus storage drive that can hold useful instructions. These instructions might include how to survive, antibiotics, or even how to glow under UV light. Added a plasmid and insert new DNA, scientists use restriction enzymes, putting DNA at specific spots. For example, one enzyme called ecoRNA because DNA at a particular sequence, leaving behind little overhangs known as sticky ends. These ends help new pieces of DNA fit in perfectly, like puzzle pieces snapping together. Moreover, we should notice that a good plasmid isn't just a ring of DNA. It needs a starting point so it can make copies of itself. It also needs a way to let scientists know if it's working. That's where selectable markers come in, like genes that give resistance to antibiotics. Some plasmids even include a gene like LAC-Z, which produce a blue color when active. This helps us see with our own eyes whether transformation has actually happened. Classic example is using a plasmid called UPC-19 with bacteria using E. coli, which was applied in my lab practice. It includes both an ampicillin resistance gene and part of the LAC-Z gene. We started by making our E. coli cells competent, meaning that they were ready to take in foreign DNA. We chilled them in calcium chloride, which helps open up their cell membranes and makes them more willing to accept new DNA. Then we introduced the plasmid and gave the cells a quick heat shock at 42 degrees. This heat shock caused temporary openings in the membrane, just long enough for the plasmid to slip in. After that, we let the cells rest in nutrient-rich growth to recover. Once they were ready, we spread them onto agar plates containing epsilon. Most cells died, but some survived. Why? Because they had absorbed the plasmid, which gives them resistance to the antibiotic. That was our first clue that transformation had worked. To take it a step further, we used something called blue-white screening. We added IPTG and X-scale to the plates. If the LAC-Z gene was intact, the colonies turned blue. If the gene had been interrupted by new DNA, the colonies stayed white. That visual difference helped us know which cells had taken up the plasmid and which had actually been modified. Inside those transformed cells, a lot was going on. The plasmid didn't merge with the cell's main DNA. It stayed separate but active. It began making copies of itself and producing proteins using the cell's own machinery. The ampicillin resistance gene made an enzyme that broke down the antibiotic. The LAC-Z gene, when working, made an enzyme that reacted with XGL to produce the blue color. This whole setup turned the cells into little factories that made new proteins, what they couldn't make before. And this wasn't just for fun. It's the same process that scientists use to create bacteria that produce real medicine in the real life. Insulin, for example, is made this way. So are some vaccines. Scientists even use transformation to engineer plants and bacteria that help clean up pollution. That's why understanding transformation matters so much. It's a powerful tool. It can help us solve huge problems. Finally, thanks to everyone who supported this journey. My instructor, Professor Lee, my classmates, and my family. And of course, I begin my appreciation to the Optimus Prime for helping make this presentation much more colorful.